Have you ever wondered how your complex thoughts, feelings, and actions arise from specific parts of your brain? It is a fascinating question that lies at the heart of understanding ourselves. Neuroscientists map brain function localization primarily by observing brain activity while individuals perform specific tasks and then correlating that activity with precise brain regions using various advanced neuroimaging techniques. This approach allows researchers to pinpoint which areas are most engaged during particular cognitive or motor processes. The core principle behind this mapping is functional specialization, the idea that different brain areas are responsible for different functions. When a specific brain region becomes active, it demands more energy, leading to an increase in blood flow or changes in its electrical and magnetic properties. Techniques like functional magnetic resonance imaging, or fMRI, detect these changes in blood oxygenation. Electroencephalography, or EEG, and magnetoencephalography, or MEG, measure the electrical and magnetic fields generated by neuronal activity, offering insights into when and where activity occurs. Positron emission tomography, or PET, uses radioactive tracers to detect metabolic activity, highlighting areas consuming more resources. In practice, this involves designing experiments where subjects perform carefully controlled tasks, such as speaking, moving a finger, or viewing specific images, while their brain activity is monitored. Researchers acquire vast amounts of data, which are then analyzed statistically to identify regions showing significant activation changes during these tasks. This process often involves comparing activity during the task to a baseline or control condition to isolate the specific brain areas involved. Advanced computational models and brain atlases help in precisely locating and interpreting these active regions. A sophisticated technique known as connectomics takes this a step further, mapping the brain's neural connections to understand how these specialized regions communicate and integrate information. Researchers also use multimodal integration, combining data from different imaging techniques, like fMRI and EEG, to get a more comprehensive picture of both the spatial and temporal dynamics of brain function. This provides a rich understanding of dynamic mapping, revealing how brain areas interact and cooperate over time, not just their static locations. By meticulously mapping brain function localization, neuroscientists gain invaluable insights into the neurological basis of human cognition and behavior. This fundamental understanding is critical for developing effective treatments for neurological disorders, engineering innovative neurotechnologies, and ultimately advancing our foundational knowledge of the most complex organ in the known universe.